7. Variation in chromosome number and structure. Genetic variation, differences between the members of the same species or those of different species. Allelic variations are due to mutations in particular genes. Chromosomal variations are essentially changes in chromosome structure or number. So this affects more than one gene. It is quite common, which is surprising. So, cytogenetic, it is a field of genetics that involves the microscopic examination of chromosomes. Cytogenesis typically examines the chromosomal composition of a particular cell or organism to allow detection of individuals with abnormal chromosome number or structure. It provides a way to distinguish between species. Cytogenesis uses three main features to identify and classify chromosomes. First is the location of the centromere, second is size, third is the bending patterns. All of these features can be seen in a karyotype. So this is how a chromosome looks like. You have the P arm and the Q arm. P is short arm, Q is a long arm. And then in between them you can see the centromere. Centromere position determines the shape of the chromosome. You have metacentric, submetacentric, egocentric, and heliocentric. Different chromosomes can be the same size and the same centromere position. So chromosomes are treated with stain to produce characteristic bending patterns so that we can tell them apart. For example, you have G bending. So in G bending, chromosomes are exposed to the dye GMSA. Some regions bind dye heavily, causes it to have dark bands. Some regions do not bind the same well, which causes it to become light bands. In humans, you have 300 G bands seen in metaphase and 2000 G bands in prophase. So as you can see here, these are the human karyotype, and the bands that you see are the G bands. So the band pattern during metaphase is different compared to the band pattern during prophase. So the banding pattern is useful in several ways. Distinguish individual chromosomes from each other, detect changes, and also reveals evolutionary relationships among the chromosomes of closely related species. Mutations can alter the chromosome structure. Structure of chromosome can be altered. The total amount of genetic information can change. So it is caused by deficiencies, duplications, but some of the mutations, it causes the genetic material to remain the same but is rearranged, like inversions, translocations. Diversity or deletion, it is the loss of a chromosomal segment. Duplication is a repetition of chromosome segment compared to normal parent chromosome. Inversion, a change in direction or part of a genetic material along a single chromosome. Translocation, a segment of one chromosome becomes attached to a different chromosome. Simple translocation, it is a one-way transfer. Reciprocal translocation is a two-way transfer. So here you can see deletion, duplication, inversion, simple translocation, and reciprocal translocation. Let's look at deletion first. You can see that the chromosome in A, you have P and Q, and then you have genes 1, 2, 3, 4, and on the P arm you have the 1, 2, 3. With deletion, what can you see? You can see 1, 3, 4 on the Q arm. Where has 2 gone? 2 has been deleted. Now we look for duplication. 1, 2, 3, 4. Now you have 1, 2, 3, 2, 3, 4. Meaning that the parts 2 and 3 have been duplicated. Next we look to inversion. C. In inversion, parts of the chromosomes are broken but reattached at the wrong order. 1, 3, 2, 4 in the Q arm. It is inverted. In translocation, it means that part of the chromosome broke off and reattached at the wrong position. As you can see, you have 2, 3, 4, but that 2, 3, 4 has reattached to another chromosome. Now that other chromosome has 1, 1, 2, 2, 3, 4. The original chromosome now only has 1, 2, 3 and 1 for the Q arm. For reciprocal translocation, it means that the chromosomes which are involved both have exchanged their parts. So you have, for one chromosome, you have 1, 2, 3 and 1, 1, 2. That's the original. But the 2, 3, 4 of that original has gone to the other chromosome, which has now 1, 2, 3, 4. So the part where the chromosomes has a change would be the 1 and 2 part for the short chromosome and the 2, 3, 4 part for the longer chromosome. They have exchanged, so that's what you call reciprocal. Deficiencies. A chromosomal deficiency occurs when a chromosome breaks and a fragment is lost. So in this picture, you can see that there's a single break. In this case, the parts 3 and 4 is lost and degraded. Then you have 1, 2, 3 and 1, 2. So it is terminal deletion. Call it terminal deletion because it happens at the end of the chromosome. Now for B, you can see that there is a break in the middle, so we call it interstitial deletion. So from 1, 2, 3, 4, the part which is 2 and 3, which is the middle, has gone. So they break, but the outer bases reattached. So what happens is that you have now 1, 4. The 2, 3 has gone. The phenotypic consequences of the deficiencies depends on the size and the chromosomal material deleted. So what was in that portion which is important to life? Are they vital to the organisms? When deletions have a phenotypic effect, usually they are detrimental, in that it can bring about very bad consequences. For example, the disease Hybride syndrome in humans. It is caused by deletion in short arm of chromosome 5. Duplications. Chromosomal duplication is usually caused by abnormal events during recombination. You have A, B, C, D, A, B, C, D. Usually during recombination, two chromosomes which are homolog, they are supposed to exchange their parts so that we can have genetic material swapping. But the thing is, even during these homologous chromosomes bivalence, there might be errors. For example, misaligned crossover. Usually, the same alleles would cross over and align with each other, B with B, A with A, C with C. But in this case, you can see that in this example, C align with B and the other C align with D. So when they align wrongly, when they do the swapping or the recombination, then the wrong parts are swapped. So instead of having the new C and D from blue, now the gray one actually has C and D, but it has two C's now, and the only one that is taken away will be the D. But in the blue chromosome, you don't have C at all because C has gone to the other chromosome. So you now have A, B, and D. C has been deleted. So there is more likeliness to have phenotypic effects if there is a large piece of chromosome being duplicated. But usually duplication have less harmful effects compared to deletion. In humans, relatively few well-defined syndromes are caused by small chromosomal duplications. Duplications can provide additional genes forming gene families. The genes in the duplicated region may accumulate mutations which alter their function. After many generations, they may have similar but distinct functions. They are now members of the gene family. Two or more genes derived from a common ancestor are homologous, and homologous genes are within a single species. They are called paralogs. As you can see in this picture, you have an open-ended wrench, but from there you can also have adjustable wrench and also a pipe wrench. All of these are wrenches. They do the same thing, which is to turn things, nuts, bolts, pipes, whatever, but they actually have their own specificity. The same goes with the gene. If a gene is duplicated, they might form a family of genes which work together and work similarly. So in this picture, you can see that a gene has been duplicated by an abnormal genetic event. Over the course of many generations, the two genes might differ due to gradual accumulation of DNA mutations. So gene number 1 and gene number 2 are mutated. They become much more different compared to the original. Originally, they were part of one actual gene. Now they have become two genes which are mutated. So we can say that they are now paralogs, which is a gene derived from a single ancestral gene. So example would be the globin genes. All of the globin genes encode subunits of protein that bind oxygen. Over 500 to 600 million years, the ancestral globin gene has been duplicated and altered. So there are now 14 paralogs in gene family on 3 different chromosomes. The different paralogs carry out similar but distinct functions. All of these genes bind oxygen, but you have myoglobin, so oxygen in muscle cells, and then you have different globins in red blood cells at different development stages. So these provide different characteristics corresponding to the oxygen needs of the embryo, fetus, and adult. So in the very early in embryonic life, you would have the myoglobin. Okay, all of these come from ancestral globin. So later in life, you would have the alpha chains of the hemoglobins. So this is expressed maximally during second and third trimester. And then you have the beta chains. Where does it come from? It comes from the same chromosome, same allele, but it is from the different dipole chain, which is the beta chains. So myoglobin is better in binding and storing oxygen in muscle cells. Hemoglobin is better in binding and transporting oxygen via red blood cells. So after birth, you have all three of them expressed: MB, myoglobin, alpha chains, and beta chains. So the majority of small chromosomal duplication have no phenotypic effect, but they are vital because they provide raw material for additional genes. This can ultimately lead to the formation of gene families. So a gene family consists of two or more genes that are similar to each other. So chromosomal deletion and duplications have been associated with human cancers, and it might be difficult to detect with carotid analysis. Comparative genomic hybridization can be used. It is developed by Anne Calioniemi and Daniel Pinker in 1992. It is largely used to detect changes in cancer cell chromosomes. Okay, first of all, isolate DNA from human breast cancer cells and normal cells. This involves breaking open the cells in isolating the DNA by chromatography. Next, number 2, label the breast cancer DNA with a green fluorescent molecule and the normal DNA with a red fluorescent molecule. This was done by using a DNA from step 1 as template and incorporating fluorescently labeled nucleotides into newly made DNA strands. Number 3, the DNA strand will then denatured by heat treatment. Mix together equal amounts of fluorescently labeled DNA and add it to preparation of metaphase chromosomes from white blood cells. The procedure for preparing metaphase chromosomes is described in figure 3.12. So the metaphase chromosomes will also denatured. Next, allow the fluorescently labeled DNA to hybridize to the metaphase chromosome. And lastly, visualize the chromosome with a fluorescence microscope. Analyze the amount of green and red fluorescence along with the chromosome with a computer. Inversions. A chromosomal inversion is a segment that has been flipped to the opposite orientation. So we start with a normal chromosome A, B, C, D, E, F, G, H, I. So you have in B, A, B, C, G, F, E, D. So the one that has been inverted will be D, E, F, G. So it is pericentric inversion where the centromere lies within the inverted region. In C you have A, E, D, C, B, F, G, H, I. So the one that is inverted will be B, C, D, E. So in this type of inversion called the paracentric inversion, the centromere is outside of the inverted region. Okay, as you can see here, you have chromosome A, B, C, D, E, F, G, H, I, which is purple. But the blue chromosome has inversion A, B, C, G, F, E, D. So to match, the chromosome which is in blue has to do a inversion loop. So A, B, C, and then the G, F, E have to make a loop so that it will match the one that is purple, which is D, E, F, G. If there is crossover happening during this prophase, what will happen is that you will have a very different type of chromosome formation. So the chromosomes which are outside A, B, C, D, F, G, H, I are normal, but a chromosome inside, because they have exchanged their parts, so now you have A, B, C, D, E, F, G, C, B, A. So the CBA is actually the duplicate of the purple ones. So what about the other one? You have I, H, G, F and E, D, H, I. So this is also a duplicate. So as you can see, the A, B, C part are now gone, so they are deleted. If you look at the paracentric inversion, which is the inversion that doesn't involve the centromere, so you have A, E, D, C, B. So again, an inversion loop happens, and if there is a crossover in this area, what you will have would be A, B, C, D, E, F, G, H, I, a normal one, and they have A, B, C, D, E, A. This chromosome has no centromere and has its F, G, H, I missing. So we call it the acentric fragment. And then the other chromosome actually has two centromeres, which is called dicentric chromosome I, H, G, F, E, D, C, B, and then F, G, H, I. So between that two centromeres, you have the dicentric bridge. Translocations. Chromosomal translocation occurs when a segment of one chromosome becomes attached to another. In reciprocal translocations, two non-homologous chromosomes exchange genetic material. Reciprocal translocation arises from two different mechanisms. First of all is the chromosome breakage and DNA repair, and second one will be abnormal crossovers. So here, in case number 1, you can see the chromosome 22 and chromosome 2. Environmental agent causes two chromosomes to break, and then you have the reactive ends. So the DNA repair enzyme recognize the breaks and they connected incorrectly. So the broken part of chromosome number 22 is bound or stuck to chromosome number 2 and vice versa. So other than that, you also have non-homologous chromosomes. Non-homologous chromosomes are not supposed to cross over. The one that is supposed to be crossover will be homologous chromosomes. But in case there is a mistake and then there is a crossover between non-homologous chromosomes, they may also exchange their parts or their segments. So now the part that is matching is now on a different chromosome. Reciprocal translocation leads to arrangement of the genetic material, not a change in total amount. So they are called balanced translocations. Reciprocal translocations, like inversions, are usually without phenotypic consequences. In a few cases, they can still result in position effects. In simple translocations, the transfer of genetic material occurs in only one direction. So they are called unbalanced translocation because one gets more information, the other doesn't get anything in case they get deleted. Unbalanced translocations are associated with phenotypic abnormalities or even lethality. For example, familial Down syndrome. So you know what is Down syndrome? It is trisomy 21, meaning that you have 3 chromosome 31s caused by non-disjunction. But in this case, for familial Down syndrome, chromosome number 21 is attached to chromosome number 14. So the individual will have 3 copies of genes found in the large segment of chromosome 31. So now you have chromosome 31, the normal one, 1, 2. But you also have another chromosome 31 segment attached to chromosome 14. So now you have 3 chromosome 31 segments. So because of that, they exhibit characteristics of the Down syndrome. Familial Down syndrome is an example of Robertsonian translocation. The translocation occurs as such. Breaks occur at the extreme ends of the short arms of the 2 non-homologous acrocentric chromosomes. The small acentric fragments are lost. So the larger fragments fuse at the centromeric region to form a single chromosome, which is metacentric or submetacentric. This type of translocation is the most common type of chromosomal arrangement in humans, approximately 1 in 900 births. Individuals carrying balanced translocations have a greater risk of producing gametes with unbalanced combinations of chromosomes. It depends on the segregation pattern during meiosis 1. During meiosis 1, homologous chromosomes synapse with each other. For translocated chromosome to synapse properly, a translocation cross must form. The matrix segregation can occur in one of 3 ways. You have alternate segregation, which is chromosomes on opposite sides of the translocation cross-segregate into cell. It leads to balanced gametes, so both contain a complete set of genes and are thus viable. Next, you have adjacent to segregation. Adjacent non-homologous chromosomes segregate into same cell. It leads to unbalanced gametes, meaning that both will have duplication and deletion are thus inviable. Number 3, adjacent to segregation. Adjacent homologous chromosomes segregate into the same cell. It leads to unbalanced gametes. Both would have duplication and deletions and are thus inviable. So in alternate segregation, you have chromosomes which are 2 normal and 2 abnormal. But you have adjacent 1, all 4 cells are unbalanced. Then you have C, adjacent 2, very rare, all 4 cells are unbalanced. What do you mean by unbalanced? Okay, supposedly you would have 2 chromosomes which is purple and 2 chromosomes which are is blue. If you look at adjacent 1, the ending will be like in one chromosome you have more blue, 3/4 blue and 1/4 purple, meaning that not enough purple. Okay, and the other also same, too much purple, lack blue. Okay. If you go to adjacent 2 also same thing, but inverted. You have chromosome number purple, there's a lot of them, but a lack of just one segment, just blue. Okay, meaning that the purple chromosome is not enough information for viability. but for the other segment, it is too much. Too much chromosome segments for viability. So alternate and adjacent one segregations are the likely outcome when an individual carries a reciprocal translocation. So they occur at about the same frequency. Adjacent to, very rare. Therefore, an individual with a reciprocal translocation usually produces 4 types of gametes. 2 viable, 2 non-viable. We call it semi-sterility. Variation in chromosome number. Chromosome numbers can vary in 2 main ways. You have euploidy and aneuploidy. Euploidy is variation on complete sets of chromosome, meaning that you should have 2 sets, 2n. Maybe you will have 3 sets, maybe 4 sets, more than normal. Aneuploidy, variation in number of particular chromosomes within a set, meaning that you have +1, -1, +2, -2, not just all set. If it is set, it is euploidy. It is just singular, it is aneuploidy. Euploid variations occur occasionally in animals and frequently in plants. Aneuploid variations, on the other hand, are regarded as abnormal conditions. In this chromosome composition of a normal fruit fly, you have diploid 2n, which is 2 sets. You have 1 chromosome for sex, and then you have 2, 3, 4. All of these are autosomes. So S2n, how many do they have? They have 8. But in this case, for polyploidy fruit flies, some of them would have 3n, which is 3 sets. So how many do they have? 3, 6, 9, 12. So they now have 12 chromosomes because they have another set. Next is tetraploid. What if they have 4 in each set instead of just 2? So we have 4, 8, 12, 16. 16 chromosomes. Now we have 4 sets, 4n. What about aneuploidy? Aneuploidy means that you +1 or -1 chromosome. So if you can see there, you have trisomy 2. So this second chromosome, chromosome 2, now has 3 chromosomes instead of just 2. Okay, so we call it trisomy 2, 2n + 1. And then for the bottom one, you can see that it is losing 1 chromosome, which is chromosome 1. So chromosome 1 is just alone. Instead of having 2, you have 1. So you can say it is monosomy 1, 2n-1. Aneuploidy. The phenotype of every eukaryotic species is influenced by thousands of different genes. The expression of these genes has to be intricately coordinated to produce a phenotypically normal individual. Aneuploidy commonly causes an abnormal phenotype. It will lead to an imbalance or amount of gene products. For example, you have 3 copies. 3 copies will lead to 150% production. A single chromosome can have hundreds or even thousands of genes. So normal individual, you have 100% of the gene product. 50% is from their father's chromosome. The other 50% is from the mother or maternal chromosome. But what if you have 3 trisomy 2 individual? So each chromosome would give 50% of the gene product. Now we have 3, so it becomes 150%. What if you have monosomy? If we have monosomy, in chromosome set, you have lost 1. So if you have 2, you have 1. So meaning that in monosomy 2 individuals, this person would have only 50% of the gene product. So these effects are detrimental. So it causes individuals to be born who are likely to survive compared to a euploid individual. Aneuploidy. They were first discovered in 1920s by Albert Blakesley and his colleagues. They studied the Jimson weed Datura stramonium. So you remember the durian looking thing? All of these 12 possible trisomies produce capsules, dried fruits, which are phenotypically different. In addition, the aneuploid plants have other morphologically distinguishable traits, including some detrimental ones. So this is the Datura stramonium. Normal one looks like that. Okay, just a plump, and then you have short thorns. In trisomy 1, you have rolled. Trisomy 2, glossy. Trisomy 3, buckling. Trisomy 4, elongate. Trisomy 5, echinus. So just like a porcupine. Trisomy 6, cocklebur. Trisomy 7, microcarpic, meaning that you can see that it is very short in thorns. Reduced, trisomy 8. Poinsettia, trisomy 9. Somehow the thorn becomes crazy long. Spinach, trisomy 10. Glow, trisomy 11. So this one bigger and longer thorns. And lastly, ilex, trisomy 12.